Yeah, welcome back. This is a third session on factors affecting our enzyme action. So already previously in two parts we were discussed these are the different factors which influencing on enzyme action. The substrate concentration is already previously we were discussed, and also enzyme concentration is also over. Then go to next slide for this next enzyme action, substrate concentration over, and effect of substrate concentration. This is what, and this is the graphical representation how to substrate concentration effect on enzyme action, and this one also. Then Km and its significance also completed. Then effect of enzyme concentration. Yeah, this is the third one, third factor which influencing on enzyme action. So that is a product concentration. So what is a product concentration? For example, if the enzyme is bind with substrate which forms enzyme plus substrate complex, and finally it turns to or which produce one kind of a product, that depends upon the substrate concentration and enzyme concentration also. Okay, then so here from the right hand side of the arrow mark, here this side, from the right hand side of the arrow mark, this side the product is continuously, yeah, I will take this pen, I can use this one. Okay, just wait. So here this is your product formation, and this is your starting material for the pathway. Okay, and here enzyme plus substrate complex formation, or whatever the intermediate which will be formed during the biochemical reaction. This is intermediate 1 and this is intermediate 2. Okay, then here this is the final product. It's a continuous process. Okay, then here this starting material which may convert into product. Then after the product formed, which is initially the substrate concentration is higher. Okay, then initially the product concentration is 0 due to there is not immediate conversion. Initially the starting material or substrate concentration is very high and the product concentration is very low. Okay, then after some time, during the chemical reaction, during the biochemical reaction, during the enzyme action, so here intermediates was formed, and after some time the product concentration slowly increases due to by the conversion of the substrate is converting, and in presence of enzyme which produce product. So the product concentration slowly increases and the substrate concentration slowly decreases. By this side, so here accumulation of this product. So immediately after formation of product, it will distribute to different tissues and different organs. Clear? The product formed which is distributed into different kind of tissues and different kind of organs. But sometimes the product concentration is increases due to over accumulation of product or accumulation of product. These accumulation of product will leads to enzyme action. Clear? So here just you read this one. The enzyme activity declines. The enzyme activity also depends on the product concentration. So that's why the enzyme activity declines when the products start accumulating into tissues and cells, or whatever the cycle where it is takes place. What kind of biochemical cycle or what kind of biochemical reaction, whichever that takes place, at that place the product concentration or the product should be accumulated. So the over accumulation of the product, which means nothing but the increase is the concentration of product. Clear? Then it will also declines or to decrease the activity of enzyme. So this is called also called as negative feedback inhibition, or sorry, simply not negative, sorry, it is a feedback inhibition, are also called as product concentration or product inhibition, right? Under certain conditions, or this is common for all biochemical reaction. Okay, so this is about effect of product concentration. And next one, coming to this one, effect of temperature. So this is also important factor which influencing the enzyme activity. For example, some of the enzymes may be having in case of if the temperature is very high, so two cases are there. One is the temperature is high and another one is the temperature is lower. The moderate or optimum rate of temperature is also that depends upon the enzyme nature. So all enzymes should be noted this one. This is the basic concept here. So here enzyme is made up of protein. So in case of if it is exposed to higher temperature, clear, the enzyme, the responsible enzyme is exposed to high temperature. So what will take place? What will happening? That is nothing but the enzyme may be denatured because it's a simply protein. Clear? So that's why raising the temperature, just see this side, raising the temperature which increases the kinetic energy of molecules up to some level, up to some extent of temperature, not complete highly higher temperatures. So here somewhat, for example 0, here the 0 degree centigrade is there, from 0 to 10 degrees it is okay, the temperature is should be within the optimum level, okay, no problem at all. Then raising the temperature which increases the kinetic energy of molecule up to certain level. So here the increasing the kinetic energy which leads to increases their motion of substrate or enzyme. Then therefore the frequency, the rate of reaction or the frequency of substrate and enzyme are the simply in terms of the collisions may be increased. Clear? Therefore the frequency with which they collide. Then this combination of more frequent and more highly energetic and productive collisions increases the rate of reaction. Okay, but here how far it is? Each and every 10 degrees centigrade. So here this is the temperature coefficient. The term we used, temperature coefficient Q10. Okay, then which is the factor? The temperature coefficient is the factor by which the rate of biological process, how much it is increases for every 10 degrees centigrade temperature. For example, the temperature raises from 0 to 10. Every 10 degree centigrade temperature will raises, and how much biological process or rate of biological process also increases. So that's why this is the temperature coefficient. Okay, look at this one. So this is for the temperatures over which enzymes are stable. Some of the enzymes are stable at particular temperatures, and the rates of most biological processes typically double. So here the rate of reaction or the rate process is double from a reciprocal, just here typically double for raising every 10 degrees centigrade. Okay, so that's why the Q10 equals 2. Okay, a 10 degrees centigrade rise in temperature will increases the activity of most of the enzymes up to how much percentage will increase the activity, 50 to 100%. Clear? This is regarding about the temperature coefficient. So this is a graphical representation. How it will be, the shape of the graph, it may be the bell shaped. Okay, then here the reaction rate increases with the temperature. So here initially the reaction rate increases with the temperature increased, the graph in between temperature and the rate of enzyme activity. Clear? And after that optimum level, the reaction rate increases with the temperature to maximum level, and then abruptly or rapidly it declines, which declines further increase of temperature. So most animal enzymes rapidly become denatured because it made up of protein at temperatures above 40 degrees. This is mandatory. Clear? Right. So these are the different effect of temperature. So the optimal temperatures of the enzymes in high organisms are rarely exceeded 50, and some enzymes lose their activity when it completely lower temperatures or frozen. Okay, then enzymes from thermophilic bacteria found in hot springs are active at 100 degrees centigrade because it is highly thermolable. Clear? Thermostability then changes the rate of enzyme catalyzed reactions that accompany rise or fall in body temperature, constitute a prominent survival feature. Cold-blooded life forms such as lizards or fishes whose body temperatures are dictated by the external environment. Yeah, this is about the temperature. Next one, come to know about pH. So what is a pH here? Then it is nothing but just hydrogen ion concentration. Okay, hydrogen ion concentration. For example, some of the enzymes highly active at particular pH. Okay, either it may be acidic environment or it may be alkaline environment or basic environment. So the rate of almost all enzymes here, this should just you look at this side. So here all enzyme catalyzed reactions exhibits a significant dependence on hydrogen ion concentration, that is especially in between 5 to 9 only. Most of the enzymes will catalyze the reactions between 5 and 9 only. That is the optimal pH for enzyme activity. Clear? Then when the activity is plotted against versus this one. So here this is pH and the rate of reaction. So this is your optimum pH range, either in between 5 and 9. Okay. Then next one, next side. So here this is also bell-shaped curve we are obtained. So just how it is, the effect of hydrogen ion concentration, how it will be influencing on enzyme activity. Just here one term we are using that. Yeah, here dissociable groups which are presenting on enzymes, right? Relationship of activity of hydrogen ion concentration and along with the between enzyme denaturation at higher or low pH ranges also. The binding and recognition of substrate molecule with enzyme, that is nothing but the active site of enzyme. The binding and recognition of substrate molecules with active site. So active site, it contains some of the dissociable groups or ionizable groups of enzymes. So that will decide what is the pH and how it will bind. The most common charged groups are there, some negative carboxyl groups and the positive charged groups, protonated amines. So these are ionizable groups which are present on enzyme. So these are called as the dissociable groups. Then will helpful to binding, or it will the binding and recognition of substrate molecules with these ionizable groups only. Clear? These dissociable groups are decide the pH range of the enzyme. So where it can survive and where at particular at which pH range the enzyme is highly active. So that will decide, depends upon the either maybe gaining or losing of critically charged groups. Yes. Yeah. Okay. This is a effect of hydrogen ion concentration pH, and this is a graph shown here. I want to show here. This one is a graphical representation. Like some of the enzymes examples are the pepsin and amidase and trypsin. One is the optimum activity for high activity than pH 2, and it should be pH 7, and this one also 8.5. Okay. Yeah. And some another last topic that is the last factor also influencing on enzyme activity. Yeah, I will continue. This is another portal. This I will continue this in another part of video tutorial. Okay, because we are having less time, only 14 minutes or 15 minutes. That is the limit of this video content. Okay, thank you.